So, good afternoon, guys. I hope I am audible and the screen is also visible to you guys. We'll be starting with the discussion of week 2 for the NPTEL course, that is Introduction to Cell Biology. And in this particular video, we'll be discussing mostly about the transcription and the translation of the particular genes. Okay, so starting with uh, So we'll be starting with the first question, that is related to the replication of the DNA. So eukaryotic organisms differ from prokaryotic organisms in mechanism of DNA replication due to the. So what can be the correct answer? So for those people who have joined just now, so we have started with the first question, and this is the question. This is related to the replication of prokaryotic and eukaryotic organisms. And so, what can be the correct answer? Okay. Any more answers? Okay. Hmm. Okay. So the correct answer is option number one and two both. Okay. So here is the explanation. So prokaryotic and eukaryotic replication. So first is, in case of the prokaryotic replication, if we see, the DNA is actually circular and it's double stranded, and single DNA molecule is present. Okay, so in case of bacteria, there is only one chromosome, and it occurs in both continuous and discontinuous mechanism. Okay, then enzymes involved are DNA polymerase 1, 2, and 3. Okay, and mostly DNA polymerase 3 is involved in the replication of the DNA. It occurs in the cytoplasm. PDF for what? Okay, so and Okazaki fragments in case of prokaryotic replication are large. Then only one origin of replication is present in the prokaryotic DNA, and then the enzyme DNA gyrase is actually needed to release the uh, release the tension created due to the unwinding of the DNA, and it adds around 2000 base pair per second, and it's a rapid process in case of the prokaryotic replication. Now, talking about the eukaryotic replication, because the DNA is linear and double stranded, what happens is uh, due to this, there is a problem that is called as the end replication problem. Okay, so because of this, the telomere that are the end of the chromosome, there is separate replication for these particular telomeres. Okay, then it occurs in the S phase of the replication, S phase of the cell cycle. Then the major DNA polymerases involved are alpha, delta, and epsilon. Okay, and out of these, because here only polymerase 3 is involved in replication, the uh, that means both the strands are replicated by this particular polymerase 3. But in case of eukaryotes, polymerase alpha actually produces the primers. Then delta and epsilon takes care of the leading and the lagging strand. Okay, so there are different polymerases that are the involved in the replication of the leading and the lagging strand. Then it occurs in the nucleus, and Okazaki fragments are smaller and multiple. Okay, here in case of the prokaryotes, it's larger, but in the other case, they are smaller and multiple. Then, if we see, the origin of replications are multiple, and then if we see there, the process in case of eukaryotes is slow. Okay, and it adds up around 100 base pairs per second because of the large number of error causing error causing polymerases. It's less speed of the replication. Okay, so if there is any doubt, you can ask. Okay, if no doubts, then we'll move on to the next question. Sorry, there is some technical glitch. Just 1 minute. Okay. So now we'll be moving on to the next part of the course, that is transcription. And from now on we'll be talking about the DNA, the expression of the particular gene. Okay, so that means we'll be talking about from now on about the transcription, and further we'll be moving to the translation of the protein, that is forming of the protein. Okay, so this is the next question. So during transcription, dash strand of the DNA is actually used as a template. So what can be the correct answer? Okay, someone said first. Any more answers? Okay, any more answers? Three. Okay, okay, okay. So the correct answer is option one, and here is the explanation. So because what we know is the replication takes place in 5 prime to 3 prime direction. Same is the case with transcription. It also takes place in 5 prime to 3 prime direction. Okay, so before going to this, first we'll have to understand that what is the coding and what is the template strand. Okay, so because there are two DNA strands, only one of these is actually being transcribed to the RNA. Okay, and RNA is actually a single stranded molecule. So out of these two strands, only one of these strands is actually transcribed into RNA. Now, if the transcription takes place in the 5 prime to 3 prime direction, then the strand that is actually running opposite to that will form the RNA in a 5 prime to 3 prime fashion. Okay, that means the DNA strand that is labeled as 3 prime to 5 prime will be used as a template for the formation of that particular RNA. Okay, so the strand of the DNA that is labeled as 3 prime to 5 prime here, that will be called as the template strand, because here the RNA polymerase use it as a template to form the complementary RNA sequence. Okay, and the other strand that is 5 prime to 3 prime strand that is there, this is called as a coding strand, because the sequence of this particular strand will be same as that of the RNA. Just the difference would be, in place of T, it would be uracil. Okay, so this is the coding strand that is 5 prime to 3 prime direction, and the other one would be the template strand. The other names for these would be, this would be the sense strand, and this would be the nonsense strand. These are the other terms that are actually being used to label these two strands, and this is the scenario. Okay, so as you can see, this is the sequence of the 5 prime to 3 prime strand, and this is the same sequence of this particular strand that is being seen in the RNA, and this is the template strand that is being used to synthesize this particular RNA. Okay, so here the answer would be option number 1. Okay, so any doubts in this case? So when, if you see a DNA strand, okay, so what happens is when there is a nucleotide, okay, so this is the sugar molecule. Here the base is attached. This is the first carbon, second, third, fourth, and through fourth, this is the fifth carbon of the sugar. This is the pentose sugar that is present in a nucleotide. Through this fifth carbon, there is a phosphate group that is attached. Okay. Now, when the nucleotide adds up to this particular nucleotide, another nucleotide adds up to this particular molecule, then the 3' OH actually forms a phosphoester bond through the other nucleotide. So this is the 5' end of the other nucleotide, 5, and this is the base. Okay, so this 3' and 5' are given on the basis of the sugar that is present here. Okay, so this particular nucleotide has this 5' end free. That means the end of the nucleotide that have 5' end free is called as the 5' end. That is why this end is particularly called as the 5'. Okay, and at the end of this strand, the 3 carbon is free to make a bond with the other nucleotide. So this particular carbon is free of that particular pentose sugar. That means its 3' end is free. That is why it is called as a 3'. Okay, so is the doubt clear? Okay, so let's move on to the next question. So if the sequence given below is the coding strand sequence of the stretch of DNA being transcribed, what will be the sequence of RNA synthesized? So this is the sequence that you have been given. So what will be the sequence of the RNA? Okay. Any more answers? Okay. 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 Uh, yeah, there is one C missing here, I guess. Yes, there is one C missing here. Okay. So the correct answer is option number one. So the correct answer is option number one. And for explaining, just now I explained, these are the two strands of the DNA, 5 prime, 3 prime, then 3 prime, 5 prime. If the RNA would be transcribed, then this strand would be the template strand, and this strand would act as a coding strand. Meaning, the sequence of this particular RNA will be same as this one. Okay, so now the strand that is given to you is the coding strand. That means the sequence of the RNA would be same as that of the disk strand, only the T in the DNA would be replaced by U in the RNA. Okay, so let's see that this would become UUCG ATGC UU A triple C then GA. Okay, so that is the sequence of the first option. Okay, is it fine? So any doubts in this particular question? Mm, okay, where are you saying? No, I think the sequence is correct, and this is the coding strand. Okay, so the coding strand would have the same sequence as of the RNA. So there is no point in complementarity here. Okay, if there would be the template strand that would be the given, then the RNA sequence would be complementary of that. So it would be the same sequence of this particular strand only. Okay, so let's move on to the next question. Prokaryotes have a dash mRNA and eukaryotes have a dash mRNA. So what can be the correct answer? Okay, any more answers? Okay, any more answers? Mhm, mhm. So the correct answer is option number two. Okay, and here is the explanation. Okay, so in case of a prokaryotic gene, what we say is there is multiple genes that are connected with a single promoter. Okay, for example, the simplest example that I can give you is lac operon. Okay, so here we can see this is the promoter of the particular gene, the regulatory gene component. Sorry, regulatory gene component. Then there is a promoter. Okay, and through this promoter, there are various genes that are connected to each other. So, in this case, there would be only one mRNA that would be produced. Okay, and this one mRNA will lead to multiple proteins. That means there are various genes that are connected through this particular mRNA, and there are multiple cistrons that are present in this particular gene. Okay, so because there are multiple cistrons here, in one particular RNA, this RNA is called as polycistronic. Okay, because there are multiple open reading frame or different protein coding genes that are present together. Okay, for example, this gene A, gene B, gene C, D, E, these five different genes will produce five different proteins, but they are connected through a single promoter. Okay, that means there are multiple cistrons that are present in this particular mRNA. Okay, so what is a cistron basically? So cistron would be the functional unit of a gene. Okay, that would actually produce the protein. So that is why this particular mRNA of the prokaryotes is called as polycistronic, because there are multiple cistrons present. Now, moving on to the eukaryotic gene structure, because in case of eukaryotes the complexity is higher, so that's why one promoter leads to only the production of one particular gene. Okay, so this particular gene would actually produce only one protein. Okay, that is why this particular mRNA would have only one cistron. So that is why this is called as a monocistronic RNA. Okay, is the difference clear between the two? So here the correct answer would be option number 2. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. Initiation of transcription by RNA polymerase occurs at which region of the DNA? Okay, any more answers? Okay, okay. So the correct answer is option number 2. This is the so whenever a transcription starts, okay, how does a transcription start? First, we have to understand the basic structure of a gene. So, in layman terms or in very simple terms, there are three main units of the gene. Okay, first is the upstream flanking region that we call it as a part of the promoter. Okay, where the RNA polymerase actually binds. Okay, now the second is the transcriptional unit, that is the region that needs to be transcribed. Okay, so this region will actually form the RNA. Then comes the third region, that is a downstream flanking region. Those regions are termed as the terminator. Okay, so these are the three main components of the gene in simple terms. Now, in case of the eukaryotes and both prokaryotes, the promoter region is actually consists of two things: the main promoter or the core promoter, and the second component is the regulatory region. Okay, in this regulatory region, there are some proteins that actually helps in the regulation of the transcription binds. For example, enhancer region. Okay, so this is the term that is used for promoter, and the core promoter is the identification site for the RNA polymerase. So the promoter region is actually identified by the RNA polymerase. Okay, and here in the figure, as you can see, this data box and the cat box, these two regions are the major regions in a eukaryotic system that are identified by the RNA polymerase. Okay, and here is the transcriptional unit, and in a eukaryotic gene, the transcriptional unit are basically divided into two parts: exons and the introns. So introns are the non-coding regions. They are actually being removed when we are forming a mature mRNA, and only the exons are present in the mature mRNA that leads to the protein after translation. And this downstream region, or the downstream flanking region, also has a terminator that is the stoppage of the transcription, that helps in the stoppage of the transcription. Okay, so any doubts in this particular question? Yeah, this is for a eukaryotic system that that figure is shown. In case of prokaryotes, there are different regions that are present there. Okay, let's move on to the next question. Which of the following property of introns in eukaryotic pre-mRNA? So, what can be the correct answer? Okay, first, okay. So, the correct answer is option number one. And so, what happens? So what happens is, in a eukaryotic system, because it's a monocistronic mRNA, and there are regions that do not code for a protein. That means these introns are actually removed during the process. Okay, so when DNA is actually transcribed, so what happens? There is pre-mRNA that is formed. So this pre-mRNA is actually a replica of the coding strand of the DNA. Okay. Now, from this pre-mRNA, to get a functional protein, we need to remove the introns. So this pre-mRNA is converted to the mature mRNA, that we call it as an mRNA, and In this case, splicing happens, that is removal of the introns. Okay, now this mRNA is mature and now it can easily go into the process of translation for the formation of the protein. Okay, so these introns are non-coding sequences that separate exons that separate exons in a monocistronic RNA. Okay, so this would be the correct answer. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. Which of the following can affect transcription? So, what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option number 5. Okay, now we'll see different options. So, in case of the chromosome location, so what happens is there is a region in the uh, nucleus that is actually called as the expression high region. Okay, so what happens is, as you know, there are two forms of chromosome or chromatin that are present. First is the euchromatin, and the other one is the heterochromatin. So euchromatin is basically a region that is high in transcription or high in expression. Okay, because this particular region is not actually wound. Okay, so it is not wound on each other, and this region is basically open in nature. That is why it is high in expression. The heterochromatin region has low expression because the chromatin is actually constricted in that particular region, and as you know, the histone proteins help in winding of the DNA. Okay, so forming of this particular chromosome leads to the more binding of this particular DNA and forming a complex structure. Okay, so whenever this particular nucleosome is formed and it is actually being converted to different forms of that particular DNA components, that forms a constricted structure. So that actually inhibits transcription. Okay, if this particular nucleosome is opened up, that means okay, So what happens is whenever we see a chromatin, so chromatin is basically the DNA that is loosely bound. When it is forming a chromosome, it needs to be wound upon itself. That means nucleosome is formed here. The histone proteins help this particular chromatin or the DNA strand to actually cover this particular length and arrange it in a form of a constricted structure, such that this particular DNA of nearly 1 meter will be arranged in a very small space. So, because of this, the DNA or the genes that are present there are actually present in a very constricted space. Okay, so that leads to closing up of the DNA. Now, whenever we have to actually express a particular gene, this particular nucleosome opens up and that helps in the expression of the genes. So that is why when we see a chromosome, if it is present in a heterochromatin region, that means it is actually have a very constricted space or a closed space. Now, if this changes to a euchromatin that is present in a open space, that means the nucleosome is actually opening up for the expression of the gene. Okay, so these two regulation happens at the level of the chromatin and the nucleosome. Now, these two, the cis regulatory elements, are basically present in the DNA itself for the regulation of the expression, that are part of the promoters. Okay, so different proteins help in actually recruiting the RNA polymerase to the DNA such that the transcription happens. So those particular proteins bind to these cis regulatory elements and help in the increase or decrease in the levels of the transcription. Okay, now the epigenetic modifications, these modifications are done when there is like, for example, there are two kinds of epigenetic modifications that happens in the DNA or in the protein. Okay, so in the DNA what happens, there is methylation that happens at the CpG islands. Okay, so CpG islands are basically CG rich regions present in the promoter. So if this CG-rich region is methylated, that means the promoter is not ready for the particular for RNA polymerase to bind to it. Okay, so that will actually repress the RNA polymerase binding to the promoter. And if it is less methylated, that means it is ready for the transcription and it will recruit the RNA polymerase. Okay, likewise, in case of the protein, these histone proteins are basically methylated, acetylated, so that they open up or they close up. That will actually lead to the regulation of the transcription. Okay, so this is how these different parts help in the regulation of the transcription. Okay, so any doubts in this particular question? Okay, so first one you understood. The second one is that whenever a DNA is actually wrapped in a nucleosome, so here the nucleosome is histone proteins are present. Now, it is actually like mm, to form a chromosome, this needs to be wrapped around itself. Okay, so that actually leads to the formation of this constricted space Okay, and this is how a chromosome is actually being formed. So wrapping up of this particular nucleosome complex upon itself and forming major structures lead to the formation of a chromosome. So now, whenever we need to have a free DNA for the expression of this particular gene, then this nucleosome needs to be unwrapped. Okay, so this nucleosome occupancy means that if the DNA is actually unwrapped, that means the gene is actually ready for the transcription or expression of the genes. So whenever this open up, that means they are actually being involved in the expression of that particular gene. Okay, now moving on to the cis regulatory elements, as we have talked in the last question also, along with the promoter, there are certain elements like the enhancers that help in the regulation of the transcription. That means these proteins actually help the RNA polymerase to bind to this particular promoter and initiate the transcription. So these cis regulatory elements are basically these enhancers that help in the binding or the initiation of the transcription. Okay. Then we have the epigenetic modifications. Epigenetic modifications are the modifications that are changed in the due course of the time. Okay. So these happens in two ways: the modifications at the DNA level or the protein level. So at DNA level, methylation is done, and methylation is generally done at the CpG islands. Okay, that means the CG rich region in the promoter. So that these regions are actually involved in the regulation of the transcription. If the methylation is more, that means the transcription levels will be less, and vice versa. Okay, so this is how a transcription is actually regulated in a whole scenario. Is it fine? Okay, so let's move on to the next question. Remdesivir, an antiviral medicine, is approved for authorized use in case of emergency to treat COVID-19 in many countries. Which step in this red numerical is related to the viral replication represented in the figure given below? Does remdesivir target? Okay. Any more answers? Yes. Okay. Any more answers? Okay. So the correct answer is option 4. That means it affects the second step, or the RNA dependent RNA polymerase. Okay. So what is actually remdesivir? It is a analog for thiamine. Okay. And what happens it doesn't have a 5 prime phosphate group. Okay. So what will happen? It is actually given in an inactive form. When it reaches the cell, it turns into an active form that doesn't have a 5 prime phosphate group. And whenever it attaches to a particular increasing RNA chain using RNA polymerase, so now when remdesivir is attached here, then it will not be able to complete its RNA replication. Okay, that's how it inhibits the RNA replication in viruses, and also it is approved in case of the emergency in COVID-19. Okay, so this is how it action helps in blocking the transcription of the particular viruses. Oh, sorry, replication of the particular viruses. Okay, so is the explanation fine? Okay, so let's move on to the next question. The formation of a lariat-like structure that is found in the top left corner, that means this kind of structure in the EM image below, is characteristic of which of the following processes? So what can be the correct answer? Okay, any more answers? Any more answers? Okay, okay. So the correct answer is option 2, that is splicing. So what happens is, whenever there is, suppose this is a RNA strand, and this is a exon 1, this is exon 2, and this is intron 1 and intron 2. Okay, now this intron needs to be excised from this particular mRNA to form a mature RNA. Then what happens is, this particular intron is removed as a lariat like structure like this, and it is actually degraded by the cell machinery. Okay, so this lariat like of structure is only seen in the splicing reactions. Okay, so that is why this kind of structure in the electron micrograph actually shows a splicing mechanism, and this is how it is how an intron is being removed from a mature from a RNA. Okay, to form a mature mRNA. Okay, is the explanation fine? Okay, so let's move on to the next question. Sorry. So now we'll be discussing in brief about the transcription process in both eukaryotes and prokaryotes. Okay, so in case of prokaryotes, there is a simpler mechanism. Okay, there is one RNA polymerase, then there is one sigma factor that actually recognizes the promoter site in the DNA. Okay, and then this helps in unwinding of the DNA and the transcription bubble is formed. So this starts the transcription process, and the first step of the process is initiation. So in initiation only, the RNA polymerase binds to the DNA and it forms a transcription bubble. Okay. Now this particular RNA polymerase moves forward and it unwinds the DNA and it also transcribes the RNA. Okay, so in the initial phase, few base pair of the RNA are actually formed. Then it moves to the elongation process in which the major protein coding region of the DNA is actually being transcribed. So here the major RNA is produced here. Okay, and that too in the 5 prime to 3 prime direction. Now, in the termination region, there is a terminator that actually helps in the releasing of the RNA from the DNA strand and the RNA polymerase. Okay, so there is a factor called as rho factor that helps in release of this particular mRNA and the RNA polymerase from the DNA. Okay, so this is how a transcription takes place in a prokaryotic system. Now, nascent RNA. Okay, so this nascent RNA is basically the RNA that is being formed from the from the particular DNA. Okay, so what happens is like there is a thing called as abortive initiation. Okay, so what happens whenever there is the binding of this particular RNA to the promoter, this RNA polymerase actually releases itself. Okay, after forming around 10 to 11 nucleotides of the RNA, this particular RNA polymerase slips off from the DNA, and this happens many times. So that is called as abortive initiation. Now, when this particular RNA polymerase is actually fits on this particular DNA after abortive initiation, then this proper elongation step happens. Okay, so that is why this particular RNA that is being produced here is called as a nascent RNA, because the system also doesn't know whether the RNA polymerase is actually being transcribing this particular DNA or RNA, or it will slip off and then again it will sit on the DNA to form the RNA. That is why a nascent RNA term is used here. Okay, so this was the prokaryotic transcription. Now we'll move on to the eukaryotic transcription in brief, although it's more complex, but I'll try to be as simple as possible. So, in case of a prokaryotic transcription, the promoter we see has many regulatory elements. In the prokaryotic system, we have the regulatory elements, but then those are less in number. Okay, so for the eukaryotic transcription, the RNA polymerase binding to the promoter is initiated by various factors called as the transcription factors. Okay, so in case of prokaryotes, we have only the sigma factor that helps in binding of the RNA polymerase. But in case of the eukaryotic transcription, there are multiple factors that are involved in the binding of the RNA polymerase to the DNA. Okay, there are, for example, this is 2F, 2E, 2H, and there are multiple more factors that are involved in binding. So, what happens here? First, these transcription factors bind to the promoter region. Then, this transcription factors actually recruits this particular RNA polymerase. Okay, so and there are various enhancer elements that, for example, this activator and the mediator, that actually helps these transcription factor to keep it binding to the DNA. Okay. Now, when this RNA polymerase binds, it starts the transcription process, and same abortive initiation already happens here. Okay, but here the like timeline for that abortive initiation is very less. Okay, now during the course of the elongation, this RNA polymerase. Okay, so this RNA polymerase has a tail, so we call it as a C-terminal tail that recruits all the enzymes that are involved in the post-transcriptional modifications of the RNA. Okay, so in case of a eukaryotic mRNA, there are three processes that takes place. First is the splicing, second is the capping, and the third one is the tailing. So splicing is removal of introns, capping is adding a 5 prime cap to the RNA, and tailing is the 3 prime end addition of a multiple A's. Okay, that is a poly A tail. So during the course of RNA synthesis, these three processes happens at the similar way, at the similar time when the RNA is actually being transcribed. Okay, so as you can see here, whenever the 5 prime end of the 5 prime end of the RNA comes out from this particular RNA polymerase, the cap is actually being attached to that particular RNA. Now, whenever this particular splicing happens, the introns are removed side by side. And whenever the termination happens, the termination is actually linked to the addition of a poly A tail. Okay, so these three processes happens in the due course of the elongation. And with the termination, the poly A tail is added to the particular mRNA. Okay, and these actually is a very complex process, and all these proteins are recruited with the help of the C terminal tail of the RNA polymerase 2. Okay, so this is the brief of both the prokaryotic and the eukaryotic transcription that we see in the system. So any doubts? I have not gone into that detail, but this is the basic system that I can explain. So any doubts in this process? Okay, so to actually save the 3 prime end of the RNA, the terminator region, there is a terminator region here also. So that actually is a poly A signal. Okay, so whenever this particular RNA polymerase reaches that poly A signal, the tail recruits an enzyme, that is poly A polymerase, that adds up the poly A tail to the that particular RNA. Okay, and due to that, the RNA is actually released from the polymerase, then it adds up the poly A tail, and then leads to the release of the RNA polymerase from the DNA also. So that is linked to the termination of this particular transcription in eukaryotes. Okay, so now the transcription is done, and then from this particular mRNA, the proteins need to be synthesized. Okay. So for the synthesis of this particular protein, we introduce now a term called as genetic code. So genetic code is a three-letter code or the three base pair code that is given to the coding sequence of the mRNA that will form a particular protein or the particular amino acid. Okay, so there, this is a three-letter code. For example, this is three bases that would code for a particular amino acid. Okay, so this combination of amino acids will form a protein or a polypeptide chain. So now we'll be starting with the genetic code and the translation. Okay, so an insertion mutation results in a DNA sequence of 49 nucleotides. So what is the maximum number of codons that can be produced by this inserted sequence? D 30. Okay, any more answers? Okay, any more answers? Okay, okay. So the correct answer is option 4. That means 16 codons will be present in this particular stretch of the DNA. Now, how? If there is any insertion mutation, so that will lead to 49 nucleotides. Okay. If one codon will be three base pairs. So if we directly divide this 49 by 3, that will give us around 16.33. Sorry, 16.33. Yes. Okay, so that means full 16 codons will be available to this particular DNA, and the last one would add up as an extra code, extra base pair. Okay, so the answer would be 60. So, any doubts in this particular question? Okay, if not, then we'll move on to the next question. Which of the following is not a property of the genetic code? So, what can be the correct answer? Okay, so the question says not a property. Okay, Okay, so the correct answer is option 1. That means the genetic code is actually universal in nature. Okay, so it will differ, not differ from species to species. If there is CAG, this would code, sorry, AUG, this would code for methionine. So this would code for methionine in every organism. Okay, so that is the nature of the genetic code, that it is universal in nature and it will not depend upon the species. Okay, so and the second one is the wobble effect. Wobble effect is basically, as you know, there would be the number of codons are 64. There are three stop codons, 61. So total number of tRNAs for this particular group must be 61. Okay, but the number of tRNAs are actually less than this particular thing. So what happens is, whenever there is a whenever a tRNA binds to this particular codon, for example this, and the opposite would be UAC. Okay, so the third one, the third base pair of the tRNA, or the first base pair of the tRNA, have a flexibility in attaching to the bases that it is attaching. Okay, in simple terms, if we see, this is a AUG that is present on an mRNA. The tRNA that binds to it would have UAC. Okay, this is from 5 prime to 3 prime direction. The opposite end would be 5 prime to 3 prime K. Okay, now this would be the first base and this would be the third base. Okay, so in case of an mRNA, this would be 1, 2, 3, and on the other side, it would be 1, 2, and 3, the other way around. So the first base of the tRNA is flexible for the third base of the codon. Okay, due to this, multiple tRNAs can bind to a particular single codon, such that multiple tRNAs binding to the same amino acid can actually be used for the formation of the protein. Okay, so this is called as a wobble hypothesis. Because of the less number of tRNAs present in the system, the the cells actually utilize this particular thing, that the first base of the tRNA and the third base of the mRNA are actually flexible and they can bind to the similar nucleotide, so that multiple tRNAs form taking the same amino acid can actually help in transcription of the mRNA. Okay, then universality I have talked about. Then degeneracy is, there are multiple amino acids that are coded by different codons. Okay, so that is the degeneracy of the genetic code. So here the correct answer would be different for every species. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. Using the codon chart given below, determine the sequence of amino acid that is produced by the translation of the given RNA strand. So what can be the correct answer? Okay, any more answers? So the correct answer is option 3. And how to see it? We have the codons and the codon table. So the codon are actually being made by the three letters. So the first letter is on the left side, the middle one is the second letter, and the third one is the last letter. And these are arranged in same order: UCAG, then UCAG, then UCAG in every third letter. Okay. So the first codon is AUG. So it would come here. That will code for methionine. Okay, then we have CCA. So C, C, and then A. That means it will code for proline. So the first one would be methionine, then proline, then CAC. So C, A, C. This would code for histidine. Then AGG. This would code for arginine. And the last one, GGA. This would code for glycine. Okay, so the correct option would be option number 3. Okay, is the explanation fine? Okay, so let's move on to the next question. Because there are 4 into 4 to the power 3, that is 64 possible combinations of triplet codons, most amino acids are specific by more than one codon. This is the concept of what will be the correct answer. Okay, any more answers? Okay, okay. So please read the question carefully. If there are 64 possible combinations of the triplet codon, most amino acids, because the number of amino acids are 20 only. So there would be cases where the amino acid can have more than one codon possible. So what this feature of the genetic code is called as. Okay. So the correct answer is option number 4, that is degeneracy. Okay, just now I explained, because there are more number of codons as compared to the amino acids. So there are 64 codons and 20 amino acids. So there would be cases where there would be more number of amino acids that would code for that was coded for more number of codons, more than one codon basically. So this property is called as the degeneracy. Okay, is it fine? Okay, so let's move on to the next question. Correct order of protein synthesis is. So what can be the correct answer? Okay. So the correct answer is option 2. That is, from DNA, first the RNA is formed. Okay. Then after formation of the RNA, splicing happens, that means removal of the protein, removal of the intron, and then mature mRNA leads to translation, that is the formation of the protein. Okay, so this is the correct order. Then let's move on to the next question. An antibiotic that specifically binds to the 40th subunit and blocks protein synthesis. Which of the following statements about action of this particular antibiotic is true? So what can be the correct answer? Okay. Any more answers? Okay. Any more answers? Okay. So the correct answer is option 3. That means the antibiotic, because it is specifically active for 40th ribosomal subunit, it will be only affecting the protein synthesis in eukaryotes. Okay. So why there is a difference in the ribosomes in prokaryotes and eukaryotes. Okay, in prokaryotes there are 70s ribosomes that have 50s larger subunit and 30s a smaller subunit. But in case of eukaryotes, the ribosomes are more in weight, that is 80s, and it has a 60s larger subunit and 40s as a smaller subunit. Okay, so if the antibiotic is actually targeting 40th subunit, that means it will be only able to target the eukaryotes, not the prokaryotes. Okay, is the explanation fine? Okay, so let's move on to the next question. Site in the ribosome from which the tRNA donates amino acid to the growing polypeptide chain is. So, what is the correct answer? Okay, any more answers? Okay, so the correct answer is option 1, that is P site. Okay, and this is a basic of the protein synthesis pathway that happens. So there are three sites on a ribosome that helps in the protein synthesis. First is the A site, then is the P site, and the last one is the E site. Okay, so A site is the attachment site where the tRNA carrying the amino acid actually attaches. Okay, so this site is the A site where the amino acid is actually attaching. Now, when this particular ribosome moves forward, there is an enzyme, that is peptidide transferase, present in the larger subunit. Okay, so that would be. Does anyone know what is the rRNA that helps in the peptide bond formation in case of prokaryotes. So, what is the name of the peptide transferase? I'm asking. Okay. So, in case of the prokaryotes, 23S rRNA that is present in the larger subunit is a peptide transferase, and in case of the eukaryotes, it's the 28S rRNA. Okay, so these two are the peptide transferase that helps in the formation of the peptide bond, and peptide bond is formed on the P side. So when this ribosome moves forward on the RNA, this peptide bond is formed on the P side. Now the amino acid actually is transferred to the first tRNA that is joined on the A side. Okay, so this particular amino acid is free. Now it will be released from this particular ribosome from the E side that is called as the exit site. Okay, so this is how a protein synthesis machinery works. Okay, so any doubt in this particular question? Okay, so let's move on to the next question. Secondary protein structure is mainly stabilized by which of the following bonds? So, what can be the correct answer? Okay, okay. Someone said covalent bond also. Okay, any more answers? Okay, okay. So the correct answer here is hydrogen bonds. Hydrogen bonds basically helps in the stabilization of the secondary structure. For the tertiary structure, it's the disulfide bonds. Okay, because the supercoiling of that particular protein on the secondary structure requires more stable bonds, that is the disulfide bonds. The peptide bonds help stabling the primary structure of the protein. Okay, and yes, so this is a scenario for the folding of the proteins and how different structures are formed. So the sequence of amino acids stabilized by the peptide bond forms the primary protein structure. So this gives the sequence of the amino acid. Based on the sequence of the amino acids, these fold upon itself to form a helix called as alpha helix and a sheet called as beta pleated sheets. These two are formed by stabilization of the hydrogen bonds. Okay, that means the amino acid fold upon itself to form helices or sheets. Okay, now these two fold upon itself more to form a more constricted structure that gives the tertiary structure of the protein. Okay, so this is actually a woolen bond. Okay, so this is the side due to the side chain interaction of the different amino acids, including the disulfide bond formed from the cysteine residues. Okay, so this is formed by the side chain interaction of the amino acid, and this is the most stable structure for monomeric proteins. That means the proteins that do not have multiple subunits. Now, these monomeric proteins are actually, if there are multiple subunits, then they form the quaternary structure of the protein that have more than one polypeptide of the amino acid, more than one polypeptide. That forms a quaternary structure. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. So omega angle of the peptide bond is acute, obtuse, right angle, or planar in nature. So what can be the correct answer? Okay, obtuse. Hmm. Any more answers? Okay, so the correct answer is option 4, that is planar in nature. Okay, and here is the explanation. So omega angle is actually the rotation angle of the peptide bond. Okay, so as you know that the amino acid structure is, okay, this is the amino acid structure, and here another amino acids form. So this kind of structure is formed. This is the peptide bond, and it has a double bond, partial double bond vector. Okay, because of the resonating nature. Now, what happens whenever this kind of structure is rotating? So it rotates in a plane. Okay, so this is a plane that is formed here. So it rotates in a plane rather than forming an acute right angle or obtuse angle. This would be a planar angle rather. Okay, so this omega angle is a planar angle. So that means option 4 is the correct answer. Okay, any doubts in this particular question? Okay, so let's move on to the next question. Which of the following amino acids are exception to the Ramachandran plot? So what can be the correct answer? 2, 3, 4. Okay, any more answers? Okay, so the correct answer is option 3. Glycine and proline have a these are actually exceptions to the Ramachandran plot because of their side chains. Okay, glycine has a only H side chain. Okay, this is the structure of the glycine, and proline is an amino acid. Okay, so proline is this kind of structure. It doesn't have a NH2 in this case. The NH is involved in the formation of this ring. So that is why it is called as an amino acid. So it, in case of proline, the rotation between the psi and the phi angles is basically very constricted. Okay. In case of glycine, it's very free. So that is why these two amino acids are basically exempted from the Ramachandran plot. This is the Ramachandran plot for glycine. Okay, and as you can see here, this blue region is the sterically allowed region, and here most of the region is allowed because of its free side chain. Okay, and this is the case for the proline, and only this much region is actually being able to be plotted on this particular graph. Okay, that is why this proline have a very constricted rotation in this case. Okay, so this is how the Ramachandran plot of glycine and proline will be plotted. Okay, so any doubts in this question? Okay, so let's move on to the next one. The rotational span of psi and phi angles in a Ramachandran plot is, so what can be the correct answer? Okay, any more answers? Okay, okay. So the correct answer is option number 3. That means from minus 180 to plus 180 is the span for the Ramachandran plot. Okay, and these are the different regions that are actually being used in the Ramachandran plot. So firstly, yes, so Ramachandran plot is the angles will that will be plotted between the psi and the phi angles. Okay, so yes, this is how it is plotted. Okay, here are the phi angles starting from minus 180 to plus 180, and here are the psi angles starting from minus 180 to plus 180. So the whole rotation from minus 180 to plus 180 would give the Ramachandran plot. Okay, the phi angles is the rotation between the C alpha and the NH2 uh, group, the bond between the C alpha and the NH2 group. The rotation of the C alpha and the CO group, C double bond O group, gives the psi angle. Okay, on the basis of the rotation of this particular thing, this depends upon the side chain of the amino acid. Okay, the side chain of the amino acid, the bulkier the side chain, the restricted the rotation. This is the case. Okay, that's how we determine whether that particular amino acid will be able to form the particular secondary structure stably or not. This is the whole purpose of the Ramachandran plot. Okay, so these are the regions that are actually being seen in the Ramachandran plot. So this region is a left-handed alpha helix. This region is between plus 60 and plus 100 of the phi angle, and the 0 and the 60 degree of the psi angle. Okay, this region corresponds to the right-handed alpha helices. This small region correspond to the pi helices, and the above region 310 leads to the 310 helices. This region leads to the beta sheets. Okay, there are different angle from which the beta sheets actually varies, and then we have the polypropylene ring that leads to in this region. So this is how we have we have to remember which region of the Ramachandran plot is actually saying which part of the secondary structure of the protein. Okay, so any doubts in this particular question? And I think I have drawn it wrong. So in the y-axis it would be psi angle, and on the x-axis it would be phi angle. Okay, so here is a correction on the my explanation part. Okay, so this was the last question for today. And if you guys have any doubt in the questions of this presentation or in any of the week's content, you can ask. Okay, okay. If no doubts, then we'll close the session for today. Thank you guys for joining in. We'll meet next week. Thank you. Thank you for joining in.